המרצה הבאה, הדוקטורנטית דאריה וג'טל. דאריה התבקשה לייצג את פרופסור רוני כהן מקנדה, שנבצר ממנו להגיע לכנס, לאור מינויו כמנהל בית חולים לילדים סיק קידס בטורונטו, קנדה. הגברת וג'טל עבדה עם פרופסור כהן במשך שנים. המחקר שלה התמקד בפיתוח קריספר, טכנולוגיה לעריכה של גנים כטיפול פוטנציאלי עבור מוטציית כפל במחלת הדושן. גברת וג'טל מונתה לאחרונה כמנהלת המחקר בחברת ביומד בטורונטו, קנדה. פליז, גברת וג'טל.
Good afternoon. So DNA is the genetic blueprint of life. Differences in our DNA is what makes you, and me, me. But also sometimes these genetic differences cause disease. And this is the case in Duchenne muscular dystrophy. Our body is made up of multiple different organs that function together collectively in our daily life. Each of these have cells inside, and each of these cells have DNA. There's about 3 billion different letters in the DNA genetic code in these cells. So as you can imagine, finding a very specific location in the DNA has been a very difficult feat. I'm showing you here muscle tissue from, at the top you can see a healthy individual, and at the bottom from dystrophic muscle. And this is a very familiar image to most of us in the room, but I just wanted to emphasize dystrophin here in red, which is that key component that is affected in Duchenne. And at the end of my talk, I will be giving one more example from another muscular dystrophy that is caused by laminin alpha-2 deficiency, and this is this protein here. So both of these examples of muscular dystrophies are affecting proteins that are key for muscle strength in our muscle cells. So I wanted to introduce an analogy. I know CRISPR is fairly new to the field, and I think this analogy can help us illustrate how it works and what it is. So if we think of DNA, which is in every single cell in our bodies, as a cookbook, then all of the genes that are written in the DNA are kind of like recipes in our cookbook. We can make multiple different types of meals based on the recipes that are in this cookbook. The next level is an mRNA transcript. So this is sort of like a chef reading the cookbook in our cells and telling what ingredients are needed to make that specific protein. So each of our exons can be thought of as a specific ingredient. DMD has 79 different ingredients. It's a pretty complex protein to make. The final outcome of the recipe for dystrophin can be thought of as the cake. And this is really what is missing in Duchenne, because the recipe is spelt incorrectly. So to sort of bring this into a context and framework using this analogy, the various different types of therapies that address, for example, respiratory care, physiotherapy, as well as corticosteroids, these are working on the level of the cake. There is no cake present, so we're in one way or another creating a scaffolding or some sort of support for our muscle cells to retain their strength and continue to be healthy. Therapeutic approaches that use utrophin, which is very similar to dystrophin, in this analogy can be thought of as, for example, providing a different type of dessert, like a cupcake, to substitute for the fact that the dystrophin cake is missing. therapeutic approaches that involve exon skipping, as we're seeing from our colleagues who gave presentations earlier. This is working on the level of the mRNA transcript. So this is altering the way that the cells are reading the recipe. And in the examples that we have right now, the technology is giving us the ability to make a partial cake. So not a full cake, but a shorter and still potentially functioning cake. Gene therapies or gene transfers, like for example, the microdystrophin examples we've been hearing about today. In our analogy, this can be thought of as adding an extra page into our recipe book. So we're not changing the recipe book itself, we're just providing a copy of the correct recipe for our cells to be able to read and make the proper cake. And again, we heard about the technical difficulties around this, and this is why in these therapeutic approaches, we are also creating a partial cake. And today I'll be speaking to you about the CRISPR-Cas9 gene editing technology specific for the population of patients who have a spelling mistake that is a duplication in their recipe. And with this approach, we're actually able to change the spelling of the recipe book itself so that the recipe is read correctly, and then we are able to create the full cake, the full-length dystrophin protein. So again, on the left panel here, we have dystrophic muscle. So there is no dystrophin expressed, and we are seeing these muscle have these hallmarks of dystrophy. And by correcting the reason why this happens, which is the spelling mistake in the cookbook, we hope to be able to turn that muscle into healthy muscle. So the CRISPR-Cas9 system is fairly new technology. It consists of two different components. The first one is the scissors, or the Cas9, and the second component is called a guide, and this can be thought of as a sort of search bar. So for example, in Microsoft Word, when you type into your find tool a particular word, this is the same thing. You type in a location in the DNA and it's able to find it. So you can search through those billions of letters of code to find a specific location. So if you design your search term or your guide to target a specific location, this brings the scissors to that location, which cuts it. The piece in the middle will fall out, and the cell, through its natural repair mechanisms, will join the two pieces together. So now let's look at, on the molecular level, what this really looks like. On the left, we have the exons from an individual that is unaffected. So we see exons number 1 through 79, all in the correct sequence. In an individual that has a duplication, they have a portion of their exons in 2 copies. So here I'm showing you a specific example of a patient who has a duplication of exons 18 to 30. So you see 1 copy here. Right after that is an extra copy, again, of 18 to 30. So this is on the DNA level, the cookbook level. On the mRNA transcript, all of the exons are placed correctly together in a healthy individual, and they are also placed together, but they don't quite fit because they're not supposed to be in that order. So in a healthy individual, they have the correct transcript and they're able to make the proper dystrophin protein. But an individual that has a duplication, they are unable to make dystrophin, and this is the problem. So this project began 5 years ago when I started my PhD in Ronnie Cohn's lab, and we were faced with this problem. So we have an extra piece of DNA in the code. So the first thing that probably comes to mind, how do we remove the extra piece of DNA, is to design 2 scissors, right? So one will cut exactly where the 2 pieces meet, so we call that the junction, and one will cut just right after. And in this way, we're removing the red piece, so we only have the gray one left. And the pros of this approach is that At that time, a couple of other labs were using CRISPR to delete pieces of the genome, and so we knew that this approach would probably work. The cons of this approach is that we require 2 different scissors, and sometimes these scissors come with unintended edits somewhere else. So if you have more than one, you have more than one of unintended edits to worry about. The other drawback of this approach is that we're really limited in where we can design our scissors. So for example, this scissor would always have to be right here. And it's not always possible to design scissors in that location. It really depends on where the sequence is. And the other drawback is that these scissors would always be specific to that one particular individual. And we wanted something that would be a little bit more universal and we could treat more than one person. So we went back to the drawing board, and what we realized is that we can actually use one set of scissors to still achieve the same thing. So this way, if you can imagine, the search term for this scissor, because it's duplicated, it will find both locations. So for example, if you were to type in a common word into Microsoft Word like the, then every single time there's a the, it will be highlighted. So the same thing works here, except our scissors are specific to those 2 locations. And so in the end, you're cutting out a piece from each section, but altogether you still have the correct number of exons and the correct number of sequence. So the pro here is now we have one guide required. So this is also good because downstream, if we're using vehicles to package the CRISPR into, then we know that we can fit one guide into a vehicle. Also, we're not really restricted in where we can design our guide because technically speaking, we can design it anywhere along the duplication. It doesn't have to be in a very specific location. And also we can use use this approach for multiple different duplications as long as they have a region that overlaps. The con of this approach was that at that time, no one had tested it, so we really didn't know if it would work, and it was a large leap to sort of see if it was feasible. So we started by treating patient cells. So these were patients, again, from an individual who had duplications of exons 18 to 30. So he had a muscle biopsy and donated his cells to our lab. And then we designed a specific guide to target his duplication and apply that to his cells. So what I'm showing you here is a Western blot. So this shows us whether or not we have dystrophin being expressed, so whether or not we have the cake. In a healthy individual, as you can see on the panel here, we have dystrophin being produced as expected. In the cells of this patient that are untreated in 3 different trials, we are not seeing dystrophin, which again is expected. This is an individual with Duchenne. Now, when we apply and treat CRISPR for the cells, this individual who previously was not producing dystrophin, the cells are now able to produce dystrophin, and this is full-length dystrophin. So this work was published in 2016, and in that publication we also looked at removing duplications for another disorder called MECP2, and we were able to show that even if the duplication is double the size of this one, which was 139 kilobase long, we were still able to remove it. So the next question now is, so we know that it works in the cells. Can we see this type of editing in a model, an animal model? So this is a long story, and I don't have, unfortunately, time to get into, but unfortunately, there wasn't a duplication model available. So the model that most labs are using, at least in mice, is the MDX mouse model, and this has a point mutation. It's not a duplication. The dog models that are being used, those also have point mutations or deletions, depending on which one we're talking about, but not duplications. So we had to make our own duplication mouse model. And so we used CRISPR to do this as well. It took about 2 to 3 years. We characterized the animal model, we performed whole genome sequencing to make sure that it in fact had a full duplication. So we were able to generate this model. and now the next step was to treat it. So before I go into that, I just want to show you, because this is a new model, what it looks like. So we can see that it is, in fact, dystrophin negative. So as you can see here, the model is not expressing dystrophin. And it does, in fact, have dystrophic muscle tissue. So we took mice from this model at 2 days after birth. And we designed, again, a guide that would target the duplication, and we packaged it into AAV9, which we have heard from our colleagues earlier today about that vehicle. And we analyzed, this project is still ongoing. The first 2 time points that I will be speaking to you about today are 6 weeks and 14 weeks post-treatment. So if we take a look at the mRNA transcript level, here I'm showing you a gel that shows us the size of the dystrophin transcript. So in a healthy mouse model, you can see the band here shows us that we have a single copy of all of the exons of dystrophin mRNA. In a duplication mouse that has been not treated, we can see that the band is taller. That's because it has those extra pieces of DNA in it. Now, in the mouse that has been treated with CRISPR-Cas9 in our new one guide approach, we can see that in the skeletal muscle, so the TA and the tricep, as well as the cardiac muscle, we are now seeing for the first time an edited transcript. So this bottom band is showing us that we have edited transcript, but we still have some transcript that has not been edited and still remains duplicated. So we have a mixture. Now, if we take a look at the actual muscle tissue itself, here in the heart and the diaphragm, in a healthy mouse, we can see dystrophin expression. We don't see it in an untreated mouse. But if we take a look at treated mice, we can see that a good proportion of the cells are now expressing dystrophin. And again, if we take a look at skeletal tissue, so for example, the tibialis anterior muscle, we are also seeing expression of dystrophin in the treated mice where we were not before. And the tricep, the expression is actually even higher than the TA. So now the question remains, how much dystrophin is enough to make a functional difference in these mice? So we performed an open field test. So essentially how this works is that you place a mouse into a chamber and that has a video camera on top connected to a computer. And the camera and the computer together work to track 100 different parameters that involve walking or pressure of the mouse paw, various different parameters like this. So I'm showing you 2 of these here. So the first one is the whole, the amount of time that is spent resting. So the healthy mouse does not rest much. The mouse that is untreated, the resting period is over here. And we can see that a treated mouse is actually performing significantly better and is resting less, so it's able to keep up and walk around. If we take a look at another measure, total distance traveled, again, we can see the total distance at the level of about 3,000 in a healthy. The untreated mouse does not travel that much, and the treated, again, is performing significantly better than the untreated. So we know that there is a functional improvement as well to this therapeutic approach. Now, before I go into sort of the take-home and summary messages, I wanted to also show you another example that we have in our lab. So as we know, DMD is caused by a spectrum of different types of mutations. I spoke about duplications, but another cohort is patients who have splice site mutations. So these are one-letter changes in the code that lead to the disorder. And there's actually another type of disorder called LAMA2 deficient congenital muscular dystrophy that is also caused by a spectrum, one of them being splice site mutations as well. This animal model, the phenotype is a lot more pronounced than we see in any of our dystrophin models. And I'm going to show you again, these mice were treated, and full length of the cake or the protein that is missing there was able to be expressed. So here we have untreated mouse. So as you can see, it has full hind limb paralysis. It's not able to stand up. And this mouse model was treated again with CRISPR-Cas9. And this is the treated mouse. It's a stark difference, isn't it? So this work was published in 2017, if anyone's interested in additional information of how this was done. So future steps. As I mentioned, this work is preclinical. We started our CRISPR program in the lab about 5 years ago, and we were able to show through the work today that we were able to develop the first duplication editing strategy to restore full-length dystrophin in patient cells and in our newly generated model of Duchenne. Longer-term experiments are currently ongoing, so we're trying to figure out things like dose escalation as well as treatment window to see what the therapeutic effect would be of treating older mice as well. So with that, I'd like to thank you for inviting me here to speak to you, and thank you to, I'd like to acknowledge the lab members who participated in various aspects of this project. Thank you. Are there any questions? Yeah. Yeah, so we are able to package both the one guide and the Cas9 into one because we're using the one guide strategy. If we were using a two guide strategy, we would need to separate them because of packaging capacity. Oh, sorry, the question was if we are using, or I guess how we are packaging the CRISPR-Cas9, if we're packaging them separately, the guide and the CRISPR, or are we packaging them together? And the answer is together for this strategy. Yeah. Phenotype? Okay. So the first question was if we had checked for off-targets, which are unintended edits elsewhere in the genome. So in the patient cells, yes, we checked using sequencing of the top predicted hits, and we were not able to identify through sequencing any off-targets. For our mouse work, that is currently ongoing, and we do plan to test. Each of these off-targets will be specific to the guide, though, so that is not to say that this will be the case for every single guide. Every guide has to be validated for off-targets. And then the second one was, the question was whether or not the phenotype of the MDX model, which is the currently sort of standard model, how is it comparative to our model? So again, that's ongoing. We don't visibly see a difference, which is the case as well with MDX in terms of the walking. It's only once we have the camera and we have a more sensitive measure do we start seeing sort of a difference. Yes? Yeah. Yeah, so we designed a whole panel of guides. That one had the highest cutting efficiency, but the benefit of our approach is that technically speaking, we could use any guide within a duplication. It doesn't have to be that particular one. So if you allow me, I want to also show this slide. So we have a panel of different patient cells in our lab. And what we're trying to do is find commonly duplicated areas. So for example, here you can see all 3 patients have a different type of duplication, but part of their intron 5 and part of their intron 6 is commonly duplicated. And we're trying to figure out how we can design guides to this area and see what the difference in editing will be for each of the different types of lengths and also locations. That's future work. Okay, thank you.
Thank you, גברת וג'טל.